Hi everyone, good evening guys. Welcome back to Unacademy NEET English channel. My name is Tina and I am your biology educator. Yes, I am back with a very important series, that is Backlog series. Many of you have started off late because of re-NEET and then counseling sessions going on, not getting anywhere, or maybe you have joined some batch late and these chapters have been left behind. So whatever be the reason for your backlog, so in this series guys, we are going to master the backlog, cover up NCERT completely. You will be making your notes with me in the class and revising things with me as well. So for the purpose of revision, for practicing, you will be joining me in the Unacademy Learners app. So make sure that you download it after the session and follow me on the profile so that we can revise a lot of questions there. All right. Good evening once again. So give me a thumbs up if you're all set with your notebooks and your NCERT so that we can start with the session. Yes, guys. So in this first session, as already told you, we will be discussing the chapter of cell, structure and function. This chapter will be discussed in two parts. So yes, quickly let me know. Are you ready? Quick guys. Are we all ready? How many parts coming up next after this? Yashu already told you this chapter will be discussed in two parts. See, I have summarized this whole chapter in just three pages, right? So you should make such short notes, not because they are short and they don't include everything. They will have everything. Every important line of NCERT will be mentioned in your notes. Is that clear? Every important word, keyword of NCERT will be mentioned in your notes. Just that you should make them precise so that, just the volume of notes should not give you anxiety attacks. Okay? And they are easy to revise also. Alright guys, so are you all set? Shall we start? So if you're all ready, so give me a thumbs up that we are ready, and also don't forget to like, subscribe and share the session with your friends. Alright guys, good evening once again. Yes, let's start this. After a long time, oh yeah. So we have been busy. All the educators have been busy in, launching new batches for NEET 2027, because of course NEET 2027 is going to be different for CBT and so many things. And also first CBT is going to happen on 30th. Don't forget that. So make sure that you join that. All right, guys. So are we all set? Shall we start with the first chapter of the series? All right guys, all right, cool. Then first thing, so I have made my short notes for your quick coverage of the backlog, so make sure you also make these notes with me. I always make my notes like this. I take a plain sheet. I'll tell you how I make my notes. So I'll always divide it. So these are my notes. Let's see how you make your notes. Let me know. All right guys, so starting with the chapter cell, the living unit of, or the structural and the functional unit of life, right? So cell is that one thing, cell is that one structure which is called structural and functional unit. Also structural and functional unit. Why? Because there is no living structure which is not made up of cell. Anything which is not made up of cell is not considered living, right? And then functional unit, because every function happening in your body is ultimately being performed in your by your cells, right? So yes, cell is the structural and functional unit of life, right? Now it helps us to differentiate between living and non-living. Living and non-living. Your NCERT calls living as animate and non-living as inanimate. So keep learning these new terms also, because ultimately we should be thorough with these terms for direct questions, right? Then now there is another difference. See, now based on the number of cells present in organism's body, based on the number of cells present in the organism's body. Organism can be unicellular or multicellular. Organism can be unicellular or multicellular, right? What is the difference? See, I've told you we are going to study everything, every bit, every line of NCERT will be covered here, okay? So you don't have to worry about your backlog now. So unicellular organism and multicellular organism, very basic. So unicellular, the single cell acts like an organism. So if a single cell has to act like an organism, therefore the single cell must carry out all the processes, right? It must carry out all the processes. Is this point clear? It should carry out all these processes, right? And therefore it helps in the independent existence of the cell. Independent existence of the cell. You can compare it with the multicellular organism. Now we are multicellular. Our skin cell cannot independently exist as an organism because all the functions are not performed. But when a cell has to act like an organism, one cell should be able to perform all the processes, number one, and it should have independent existence. Please let me know that this point is clear to all. Is this point clear? While multicellular organisms, body is made up of multiple cells or many cells, multiple cells. And when the body is made up of multiple cells, then there is an advantage also. The advantage is now cells can show division of labor. One cell doesn't have to do it all. Are you getting it? So there is division of labor. It leads to division of labor, and that means the tasks are divided. Now advantage number two is whenever the task is divided, it always leads to specialization, right? So division of labor, which leads to differentiation or specialization. Is that clear? It leads to differentiation or specialization. Please tell me these points are clear. Anyone with any doubt? Yes, please. Anyone with any doubt? This is Phoenix 3.0. Yes, Tamil, this is the same ma'am who's teaching in Phoenix 3.0. So as said, yes, I have already discussed this chapter in the batches. But those who have missed out those chapters or other students who have not joined the batches yet, this is for all of you. Is that clear? Good evening, guys. Good evening. So by the way, please don't get late. These classes will be 3 to 4 times a week because I've told you how to cover the backlog. The rule number 1 is the ongoing syllabus should always be your priority and you have to extract time to cover up your backlog. So we are extracting time, just 3 to 4 classes in a week, and rest of the week you can cover up your entire other syllabus, right? And these classes will be for 1.5 to maybe less than 2 hours, right? Is that clear? Okay. So unicellular and multicellular, all done. Now moving on to the next, that is cell theory. Let's talk about some history here. So, okay, let me give you the historical background. The historical background of these discoveries. Yeah. Point one, the first cell was discovered. Yes, we all know that. First cell was discovered by Robert Hooke. Yes, first cell was discovered by Robert Hooke. Now the cell that Robert Hooke observed and discovered, that was actually a dead cell. That was actually a dead cell, and it was nothing else but cork cell. Now, what he observed was actually like a honeycomb, this cork cell. So cork, so these are cork cells. These cork cells were arranged in this honeycomb-like structure, and they were empty. They were empty. So these were empty spaces. These were empty compartments. And these walls that he observed are actually cell wall, because it's a dead cell. So all that you can see is just cell wall, right? Otherwise there is loss of protoplasm, nothing, no living fluid. So compartments, and these compartments are actually called cells. So what he observed was a dead cell, a cork cell with honeycomb-like structure. Honeycomb-like structure. Noted? Then first living cell. The first living cell was observed by Leeuwenhoek. Anton von Leeuwenhoek. He observed the first living cell. Which cells did he observe? He observed bacteria, RBC, human RBC, okay, then human sperm, then many protists like, amoeba, protozoans, amoeba, paramecium, all these protozoans were observed by Leeuwenhoek by his self-designed microscope. Is that clear? Then another historical background. So many scientists will keep coming along with the chapter as the chapter proceeds. Then coming on to cell theory. So gradually came up cell theory. The cell theory was proposed by two scientists. One was Theodor Schwann and Matthias Schleiden. Right? Matthias Schleiden. So basically, Matthias Schleiden was a German botanist. Sorry, you have to remember that, yes, he was a botanist, both German, and he was a zoologist. Both German. Is that clear? And the cell theory was proposed in which year? Yes, guys. Cell theory was proposed in which year? In 18. So Schleiden gave his part of the theory in 1838, and he proposed it in 1839. So overall we call it 1839. Right? Now let's discuss the cell theory in detail, and later on we will see that it will be modified also. It will be modified by Rudolf Virchow. Now what was the modification? Why was there a need of modification? We'll talk about it all, but this came in 1855. Are these points clear? Yes, are these points clear, everyone? Now let's see what each one of them has proposed. So in cell theory, see here. Yeah, first person was Schleiden, the botanist, right? What did Schleiden observe? So Schleiden had studied lots of plant cells, lots of plants. So Schleiden said that I have observed so many plants, and all plants are made up of cells. So Schleiden proposed that all plants are made of cells. So being a botanist, this is what he proposed. Is this clear? Botanist is someone who studies plants. Next scientist was Schwann, the zoologist. So Schwann observed that all animals are also made up of cells, but other than this, he also proposed certain other points. Now see, Schwann said that all animals, number one, all animals are made up of cells. That is one. Then, because Schleiden was a botanist, he had studied plants, he observed that there is a cell wall, but he didn't mark it like it is only present in plants, right? But when Schwann compared his observation with what Schleiden had already proposed, Schwann said that there is that cell wall is only unique to plants only. Sorry, only. It is unique to plants. Is that clear? It is unique to plants, and the thin outer layer, there is a thin outer layer around all cells which is called plasma membrane, which is known as plasma membrane. Is this point clear to all? Please tell me, guys, are these points clear? Are these points clear? Hi, Tarunika. Yes, I remember. It's been a long while. How are you and what are you doing these days? Yes, guys. All clear? Cool. And then we have third. So this is what was proposed by Schwann and Schleiden. Is that clear? Now, do you see neither of the two have told us how the cells are born, how cells come up, how new cells are produced. So there was a need to modify it. There was a need to slightly modify it. See, this was the original cell theory. But it was modified. It had to be modified by Rudolf Virchow. What was the modification actually? The only modification needed was to mention how new cells are generated. So he explained how new cells are formed, explained how new cells are produced or formed. And what did he say? What did Rudolf explain? He said cells come from pre-existing cells. Cells come from pre-existing cells. But what exactly he stated? Cells come from pre-existing cells. The way he exactly, the way he exactly said it was omnis cellula a cellulae. Omnis cellula a cellulae. Is this point clear? This is what he said. Omnis cellula a cellulae. And you have to remember that who gave the cell theory and who modified it. So it should be crystal clear to all of you. Anyone with any doubt, please let me know. Is that clear? Is that clear? Noted? All right. So this was about cell theory. Now, so basically, overall, Schwann was the one who stated. So there is this NCERT line, guys. You have to put it in a box while making your notes. Write down that bodies of plants and animals. Bodies of plants and animals are composed of cells and their products. This line was said or given by, yes please, by Schwann. Is that clear? This was said by Schwann. NCERT. Always write down direct NCERT lines, important lines in a box and write down particularly NCERT so that you know this line is important. Is this point clear? So you have to take out important statements like this. Yes, guys, are these points clear to everyone? Right? Now, let's have an overview. Yes, guys, let's have an overview of a cell. Yes, please. See, by the way, cells come in different sizes and shapes. So if I have to give you some idea about the size of different cells. So different cells, please note down size of different cells, and then I'll talk about the shape also. So for example, the smallest cell which is mycoplasma, the size of mycoplasma is 0.3 micron meter. We will not talk about a virus here. Virus is not a cell. Are you getting that? So smallest cell is mycoplasma, size is 0.3 micron meter. Size of bacteria as given in the theoretical part, it is 3 to 5 micron meter. But in the diagrammatic form, they have given a different value, that is between 1 to 2. So both are right. Both are right because bacteria come in many sizes, right? And then human RBC. The size of human RBC, as mentioned, is 7 micron meter. Now the largest cell is ostrich egg, which is visible to the naked eye. So it's big, quite big. One egg is enough to make the omelette for the whole family. That is the size of the ostrich egg. Noted? Then coming on to different shapes. Now see, the shape of the cell is actually the function, is dependent on the function it has to perform. For example, you see longest cell is your nerve cell. Now nerve cell is highly branched and it can go up to 1 meter of length. Not all are, but nerve cell or neuron can go up to a meter, and it is branched and it is elongated. Is that clear? Then if we talk about RBC has to be very small and has to have a shape with large surface area. So it is discoidal. This shape has very large surface area to accommodate more of hemoglobin, to discoidal. Are you getting it? Then as far as muscle cell is concerned, depending upon, let's say, smooth muscle is spindle shape, showing contraction. Are you getting this? So the shapes depend upon the function that a cell has to perform. Noted? All right. Now let's get an overview of the cell. Let's take an overview of the cell. What are the basic components of a cell? Components of a cell. Is that clear? What are the basic components of a cell? Now listen, any cell, I'm not talking about prokaryotic or eukaryotic, you all have heard of, right? I'm not talking about that. Any cell, in general, any cell will have these three components. What are the three components? Number one, it will have a cell boundary. It will have a cell boundary, right? Then cytoplasm. Cytoplasm is that matrix where all the cellular activities will occur, and then it will have a genetic material. So when these three components combine in a particular way, they form a cell. So every cell should have these basic components. You will come across certain cells where, let's say, the genetic material is not there, nucleus is absent, right? You know some examples, but don't worry, it's not that they were never present. Nucleus was present maybe in the precursor cell, but gradually, in order to accommodate a particular function, it lost certain structures. Well, that is called an adaptation. Otherwise, a typical cell will have all these three components. What is? One, boundary; second, cytoplasm; and third, the genetic material. Are these points clear? Anyone with any doubt? Is that clear? Noted? Right? So yeah, if we talk about the cell boundary, guys, the cell boundary can be of This boundary is made up of different components. It could be cell wall, right? Then plasma membrane, cytoplasm. Cytoplasm is always a semi-fluid matrix, where cellular activities occur, where, or as NCERT says, main arena for cellular activities. Is that clear? And the genetic material. Now genetic material is generally DNA, right? And this DNA is present in the form of chromosomes. Number of chromosomes will vary from each species to species, right? But what is that chemical which forms the genetic material? That is DNA. Are these points clear? So these are the three typical components of each and every cell. Please tell me these points are clear. Nothing very heavy, right? Is this point clear to all? Noted? Now, there is this line in NCERT, guys. There is this line in NCERT. Note down very carefully that chemical reactions, written, listen, chemical reactions occur in it to keep the cell in living state. Is that clear? So this is direct NCERT line. You must write and put it in a box. Clear? Write down NCERT. This is direct NCERT line. Now you should know that these chemical reactions that we are talking about, all these chemical reactions occurring in the living cell, these are known as metabolic reactions. Yes or no? I hope we know this. All these living reactions are called metabolic reactions. Anyone with any doubt, quickly let me know. Yes, please. Yes, main arena for chemical reactions to keep the cell in the living state. Noted? And if you remember a little bit about that first chapter, Living World, metabolism is a defining property. Without these chemical reactions, cell cannot be living. In fact, nothing can be marked as living without these metabolic reactions. So hashtag metabolism is the defining property. Are these points clear? Anyone with any doubt? Yes, please. Noted? One more thing. NCERT has called a particular membrane as a delimiting membrane. So they have used this term delimiting membrane for plasma membrane. Delimiting membrane. See, cell wall is not present in all. We know this. Delimiting membrane. But plasma membrane will be present in all the cells. Is that clear? Plasma membrane will be present in all the cells. Cell wall may or may not be present. Clear to all? Is that point clear? Noted? Now, on the basis of this genetic material, we can classify cells into, see, on the basis of this genetic material, how it is packed, where it is present, we can classify cells into two types. One is prokaryotic and second is eukaryotic. Please tell me these points are clear to you. So there are prokaryotic cells, there are eukaryotic cells. The difference is on the basis of how this genetic material is present in the cell, and that's our next point of discussion. Yes, guys, quickly let me know. Anyone with any doubt here? Shall we proceed? So let's see the basic difference between, first let's see the basic difference between a prokaryotic and a eukaryotic cell. Prokaryotic and a eukaryotic cell. Prokaryotic and eukaryotic cell. Yes, please. So as the name itself says, I'll break the word prokaryotic, eukaryotic, right? So what are pro and what is pro and eu? Otherwise, pro means, expert. But here, whenever we are talking about science in biology, right? So in the world of biology, pro means primitive. Carry-on word represents nucleus. So primitive nucleus, right? While EU stands for true. Carry-on is again nucleus, true nucleus. So what do we exactly mean by primitive nucleus? Let's have some more discussion. See, in a prokaryotic cell, the genetic material is lying freely. Let's say this is a cell, right? Let's say this is the plasma membrane. This is all cytoplasm. And now the genetic material is lying freely in the cytoplasm. This is the genetic material. So this is the difference number one. Genetic material lies freely in the cytoplasm. Is that clear? There is no compartmentalization. Why it lies freely in the cytoplasm? Because of no compartmentalization. Like, it's like a startup. It always starts with one room where everybody is sitting, discussing, and then everything is happening in that one room, right? So this is the beginning of life. This is how the cell became, because of no compartmentalization. It is the beginner's level. Is that clear? Not only that genetic material is lying freely in the cytoplasm, but in fact, the whole structure is very, very simple, very basic structure, very simple structure. There are no specific, no specific structures for different functions. So, no membrane bound organelles present, no membrane bound organelles, no membrane bound organelles. When it's a startup, everybody has to do everything, otherwise not possible to grow. So in the beginning, you will see that in the prokaryotic cell, the plasma membrane will, function in different ways. It will act like a mitochondria also. It will store pigments also as and when needed, right? And of course, it is the delimiting membrane. But it will not have separate organelles like mitochondria for energy production, chloroplast for photosynthesis. No, it's not happening yet. Is that clear? So, no membrane bound organelles like mitochondria, ER, Golgi bodies, right? All these structures are absent here. Even the ribosomes are of very small type. So ribosomes are also small and we call this type as 70S. What does 70S exactly mean? We'll discuss under the topic of ribosomes. Is that clear? But they are small, they are primitive type. Please tell me these points are clear. Now what is all included under the category of prokaryotes? So when we say prokaryotes, it includes your blue-green algae, right? Then it includes bacteria, the eubacteria, right? Then mycoplasma, eubacteria as well as archaebacteria, mycoplasma and PPLO, pleuropneumonia like organism, all are included in the category of prokaryotes. Is that clear? So these are the organisms. Now coming to eukaryotes. So unlike prokaryote, here you will see there is proper organization. So we have a separate chamber for nucleus, right? For genetic material. And there are other components also which have developed. So genetic material lies in this chamber. instead of just lying randomly in the cytoplasm, genetic material is membrane bound, is membrane bound. Now, they've grown richer, developed. So there is compartmentalization. So it is membrane bound, there is compartmentalization, rather different works are occurring under different compartments, compartmentalization, right? So there are membrane bound organelles here, membrane bound organelles present. Which are the membrane bound organelles? We have already told you, mitochondria, ER, Golgi bodies. We'll discuss each one of them gradually, right? All these structures are present. Is that clear? Chloroplasts, that means plastids, all present. Anyone with any doubt? Noted? Noted? As far as ribosomes are concerned, you will see in eukaryotes, yes, you will see the presence of 70S ribosomes also at certain places. But then, according to the evolution, as per the evolution, bigger ribosomes have also developed, which are marked as 80S type. So we see both types of ribosomes in eukaryotes. Is this clear? So we'll see different boundaries of mitochondria, ER, Golgi body, lysosomes, chloroplast, and so many things here. Is this clear? Now, what all comes under this eukaryotes? So we have protista, fungi, right? All the fungus comes in here. All animals, all plants, algae, not blue-green algae. Blue-green algae is basically a prokaryote. Otherwise, all green algae, red algae, brown algae, unicellular, multicellular, all comes under the category of eukaryotes. Please tell me these points are clear to everyone. Is that clear? Noted? Anyone with any doubt? Yes, guys? So if this is all clear, quickly share. Everybody has to bring at least, one friend along. Let's go and attend Tina Maam's, backlog class. I'm sure most of you are facing this backlog problem. This ideal situation of, everyone totally in sync with the ongoing syllabus, it's rare. It should be possible, and that's why we are here with the backlog series, so that we can quickly cover up the backlog, right? And also, I have not started with any course for NEET 2027 on YT. So here it is. All right, guys. Is that clear? Everyone? Now, moving on to the next topic. The next topic is the exact, so we will be discussing about the prokaryotic cell in detail. Let's talk about the prokaryotic cell. Yes. Prokaryotic cell. Yes, please. So just like any other cell, I have said, any cell will have these three components, but there will be differences, of course, in prokaryotes and eukaryotes, and then in every eukaryotic cell also there will be differences, but these are the three basic things which any cell should have. So let's see. So prokaryote, a prokaryotic cell will also have these three components. It will also have these three components. Number one is a cell envelope. So the boundary is known as cell envelope. Noted? Cell envelope. Clear? Yes. Second is cytoplasm. Second is the cytoplasm, where actually the reactions or the activities will occur to keep the cell living. Noted? And then comes the genetic material, based on which we have marked it as a prokaryote. Genetic material. Anyone with any doubt, please let me know. Yes, guys. These three components all well clear and well acquainted with the terms, right? Now, let's talk about cell envelope. Cell envelope. First, I'll write down what comes under each category, and then we'll discuss each one of them in detail also. So, cell envelope consists of three layers basically. It consists of three tightly bound layers. There is no space in between. 3 tightly bound layers, right? Which are, number 1, we have glycocalyx. Second is cell wall, and third is plasma membrane. All three of them are intact and collectively known as cell envelope. Is that clear? Now, second is cytoplasm. Well, we have already discussed this is semi-fluid. Whether it is a prokaryote or a eukaryote, cytoplasm is always semi-fluid matrix. Okay? And it is a main arena for all the cellular activities. By the way, this cytoplasm also has two components. One is cytosol. Now, cytosol is actually the matrix part, and within this matrix are, present organelles. I understand prokaryotes don't have many organelles except for ribosomes, but whatever, all those structures are present in the cytoplasm. So the only organelle present here is ribosome. Is that clear? And then there are some inclusion bodies. These inclusion bodies are not found in eukaryotes. They are characteristic of prokaryotes. So these structures are also found in the cytoplasm. Noted? We'll discuss each one of them. And then finally, we have genetic material. Yes, this genetic material, as I said, it is freely lying in the cytoplasm. It is lying freely in the cytoplasm. Is that clear? And so, no well-defined nucleus. No well-defined nucleus lies freely in cytoplasm, and this area, this region where it, as far as this genetic material extends, that region is known as, nucleoid. Region is known as nucleoid. So it doesn't have a nucleus. Instead, what does it have? It has a nucleoid. Are these points clear? Noted? Now we'll come back to it gradually, but this is the basic of, like, overview of a prokaryotic cell. I should tell you right here. So genetic material is one, present in the form of nucleoid. There is some extra genetic material also present in the form of, extra chromosomal genetic material is also found here in the form of plasmid, right? So these two structures come under the category of genetic material. Anyone with any doubt? Yes, please. So the quick overview is clear. Cell envelope made up of 3 tightly bound layers. Now let's see the details of each. First of all, glycocalyx. See, glycocalyx may or may not be present. So that's why I'm writing plus minus. Cell wall also may or may not be present. Mostly present, but still exceptions are there. Plasma membrane will always be present. Is that clear? First, let's discuss more about cell envelope. Come here. So cell envelope. So cell envelope consists of, as said, how many layers? 3 tightly bound layers. There is no gap in between. Okay, there is no gap in between. And first let's draw this only. So first, the outermost is glycocalyx. Then we have cell wall, and the innermost is plasma membrane. Innermost is plasma membrane. Glycocalyx, cell wall, and the innermost being plasma membrane. So you must write these points. The sequence, outside to inside. See, this is the outermost. If all 3 are present, then this is the outermost, this is in the middle, and this is the innermost. Is that clear? Obviously, if glycocalyx is not present, then this will become the outermost layer. So just use your brains. All right. As said, this may or may not be present. This may or may not be present. This is always present. Okay. Now, first of all, what does this glycocalyx do and where is it present? See, by the way, okay, glycocalyx, let me first draw it for you. So, let's say this is a bacteria, right? And this one, you can say the innermost layer, this is the cell wall, and then we have this layer called plasma membrane. Now I have kept some distance in all these layers just to keep it clear, okay? Otherwise there is no gap in between, they are tightly packed. All right, so this red one is the glycocalyx, this one is the glycocalyx, the yellow one is a cell wall, and the white one is the plasma membrane. Are these points clear? So I hope the location is clear to you. So outermost, middle and innermost. So what does this glycocalyx do actually? First of all, this glycocalyx is meant for, it helps the bacteria to hide from host's immune system. Clear to you? It helps the bacteria to hide from the host's immune system. Noted? Now, second, now this glycocalyx may be present in two forms. So, first of all, it may be present, it may not be present. May or may not be present. Okay. So, if it is not present, listen here, if it is not present, such bacteria are known as R strain. Now listen, if this glycocalyx is not present, it will not be able to hide from the immunity. Let's say a bacteria enters my body. My immune system is active 24/7. So, my body, my immunity will only be able to see that bacteria which does not have glycocalyx. Do you understand that? So, R strain will be visible to my immune system. My immune system is active, strong enough, it will destroy the bacteria. So, that bacteria will not be able to cause disease. So, R strain is not disease causing, we call it non-virulent. R strain. Is that clear? Then, if it is present, then in that case, it is known as S strain. Now, which is not visible to the immune system, will be able to cause the disease. Hence, S strain is virulent in nature. It is virulent in nature. Is this point clear? This one is virulent. Noted? Third point about it is glycocalyx can be present in the form of, number one, capsule or a slimy layer. What is the difference? See, if the glycocalyx is loose and slimy, then we'll call it slimy layer. And being slimy, slime is like, the mucilage like appearance. So it will also help in attachment, of course. And if it is tough and hard, then it will be known as capsule. Is this point clear? So when do we call it capsule and when do we call it slimy layer? Are these points clear to you? Please tell me. This is all about glycocalyx. Yes, everyone. Clear to all? Noted? right? Cool. Then coming on to the middle cell wall. See, first of all, the cell wall determines, as always, the function of cell wall is, wait, I'm rubbing this off, okay? All right. So what is the function of cell wall? The function of cell wall is to provide shape, rigidity, and also this is one, this is two. It provides shape, rigidity, and it prevents the cell from bursting. Prevents the cell from bursting. Are these points clear to everyone? Done? Then, what is this made up of? What is it made up of? Listen. It is made up of mainly amino acids plus sugar. Amino acids plus sugar. So we call this peptidoglycan. We call it as peptidoglycan. Amino acids plus sugars. Is this point clear? Now, as I said, it may or may not be present, right? It may or may not be present. For example, it is absent, it is present, right? Now, similarly, where is it absent? Mostly present, but it is absent in, see, absent in mycoplasma. Absent in mycoplasma. Is that clear? So, because the function is to provide shape and rigidity, so it means mycoplasma will not have fixed shape. Therefore, no fixed shape. That's why it is known as the joker of plant kingdom. Is it clear? So mycoplasma has no fixed shape. It keeps on changing its shape. Pleomorphic. Anything which can keep changing its shape is called pleomorphic. Is this clear? Now, are these points clear? Then, third point is about plasma membrane. So the function of plasma membrane is to, because it is selectively permeable. See, I'll tell you the basic nature. Hashtag, this one is freely permeable. So it will not check which molecule is entering or leaving. That is not the function of cell wall. That is the function of plasma membrane. So it is selectively permeable, and that is exactly the function also. It will keep a check. Selectively permeable. So only those which are allowed by the plasma membrane can enter or exit. Is this clear? Now the structure, overall structure will be similar in both, overall structure is similar in prokaryotes and eukaryotes. So I'll talk more about plasma membrane in eukaryotes. Similar structure in pro and eukaryotes. Okay. So we'll not talk about the structure exactly here, but in general, they're made up of proteins, lipids, carbohydrates, just as it is in that. Clear? Now, I have mentioned already that prokaryotic cell is like a startup, right? It's at the beginner's level. So there are no different specialist structures like mitochondria or chloroplast and so on. So plasma membrane itself is carrying out different structures, different functions. And in order to carry out those different functions, it will show certain modifications also. Let's talk about the modifications here. So the modifications of plasma membrane. See, modifications of plasma membrane. Number one is mesosome. Number two is chromatophore. And number three is pili or fimbriae. Is that clear? So these are the general things that are present in here. Please tell me these points are clear to all. Yes, please. Please tell me these points are clear to everyone. Done? So these are all modifications. Now what are these modifications meant for? Let's understand. First of all, I'll talk about mesosome. So these are modifications of plasma membrane, PM, modifications in prokaryotes. Is that clear? So first of all, let's talk about mesosome, right? So mesosome, how is the modification like? See, let's say this is the plasma membrane, right? So plasma membrane can fold inwards like this. This is plasma membrane. Folding like this. Infolding of plasma membrane will form the mesosome. So what is mesosome? Mesosome is the infolding of plasma membrane. Is that clear? That is how it is formed. It can be in this form, it can be in the form of like sac-like structure or a tube-like form. So mesosome can occur in either way. This one is known as lamellar. This one will be known as sacular, sac-like, while this one will be known, or this one can also be known as vesicular, and this one is known as tubular. Okay, so mesosome can be in any, the infolding can be in any of these forms. Noted? Anyhow, it is increasing the surface area. So what is the function of infolding? So infolding is meant to increase the surface area. Is that clear? This is to increase the surface area. Please tell me these points are clear to everyone. This is to increase the surface area. Anyone with any doubt here? Noted? Now, what are the different functions? That is very, very important. So, what are the functions of mesosome? Now, function number 1, because it is increasing the surface area, there is some enzymatic activity occurring here. Enzymatic activity. Point number 2, this mesosome, because of the infoldings, increasing the surface area, helping in enzymatic activity, it is going to act like the mitochondria of bacteria. Is that clear? So, it helps in respiration, energy production. Clear to all? Then third function is secretion. Also, it will help in cell wall formation. 5, it will help in DNA replication. Not only replication, but its distribution to the daughter cells also. And that's it. Distribution to daughter cells. Noted? Therefore, now because of the function, this function, we can say mesosome hashtag is analogous. Analogous means performing similar function, analogous to mitochondria. Are these points clear to everyone? Yes, guys. Anyone with any doubt? Monocotyledonous root. What is monocotyledonous root doing here? The brain is flipping from one place to another. How will you focus? Now, guys, next is chromatophores. Yes, the second modification I have written is chromatophores. What is the function? Let's understand. See, of course, you know chroma. Chroma stands for pigment, color. Phore means bearing. So this is such modification which bears pigments. So it is a pigment storing modification. Pigment storing. So pigments are always meant for light absorption. Therefore, it is present only in those bacteria which are photosynthetic. Present only in photosynthetic bacteria. Because you require pigment only for light absorption. Photosynthetic bacteria. Is that clear? Like blue-green algae, like that. So how is that So instead of this plasma membrane, now plasma membrane will again infold like this, like this, like this, like this, and so on. And within this, there are pigments stored. These are chromatophores, and on the outer side you have the cell wall, and glycocalyx may or may not be present. Please tell me these points are clear to everyone. Anyone with any doubt, please tell me. Noted? And then talking about the third modification, this third modification is known as pili or fimbriae. Now see, pili and fimbriae are meant for attachment. They are not meant for attachment. They have no role in locomotion. For attachment to the surface or to the other bacteria or the host. Surface, other bacteria or the host. Is that clear? But the main thing is, it is meant only and only for attachment. Hashtag no role in motility. For movement's sake, bacteria should have flagella. Is that clear? We'll discuss that as well. Now, what is the difference between fimbriae and pili? Now, pili, the name is small, but it is a bigger structure. It is long and tubular. It is long and tubular, right? Longer in size, and this one is smaller in size or shorter in size. Bristle-like. Is that clear to everyone? Please tell me these points are clear to all. Also, you will see, so it is meant for attachment to other bacteria. Why does it have to attach to the other bacteria? For conjugation. Now, conjugation is a type of sexual reproduction as seen in bacteria. So, during this conjugation, they exchange their genetic material. Okay. So, in order to exchange their genetic material, this pili, therefore, pili will form a conjugation tube. Is that clear? It will form a conjugation tube like this. Let's say this is one bacteria, this is another bacteria, and this one will give off pili like this. This pili will attach to the other one, and the walls will merge to form the conjugation tube. Please tell me these points are clear. Anyone with any doubt? Yes, please. Noted? Now, coming on to the next part, which is flagella. Now, flagella is not present in all. Flagella will only be found in those bacteria which are motile. So, the motility is taken care of by flagella. This is for motility. This is for motility. Motility means for movement. Any flagella, even flagella will be found in eukaryotes. Any flagella is made up of three parts. One is basal body, then hook, and the filament. So filament is a long thread-like part, right? This is a long thread-like, which will actually wave, right? This will move. Now these are the three parts, and it is made up of a protein called flagellin. See, the eukaryotic flagella is of different protein made up of flagellin. Structure of eukaryotic flagella is also in your syllabus. We'll discuss it. It is quite complex, 9+2 arrangement, right? This is made up of simple protein called flagellin. Are these points clear to everyone? Is that clear? Done? All right. So what does NCERT write about it? So NCERT says it is thin, it is filamentous extension, thin filamentous extension through cell wall. So remember, what has NCERT written? It has thin filamentous extension through cell wall, not plasma membrane. So of course it will come out through plasma membrane and finally out of cell wall, but note down this word. Is that clear? Note down this word and put it in box and write down NCERT. Is that clear? Through NCERT. So cell wall word has to be remembered. Clear to all? Done? All right. So after flagella, we are done with the cell envelope. Okay, so flagella is not a part of cell envelope, of course, but I've just mentioned it because it will be helping in motility. I wanted to compare it with pili and flagella. Pili and fimbriae. Flagella is an independent structure. So cell envelope is clear, right? There is one more thing about cell envelope. So bacteria can be divided into two different categories on the basis of some differences. We have this technique called gram staining technique, which will divide bacteria into gram positive and gram negative. Okay, so that technique is certainly there. Now talking about cytoplasm. Okay, so about cytoplasm, I've already told you it is semi-fluid, and there is jelly like material called cytosol, and it has just two components, which is called ribosomes and inclusion bodies. So ribosomes are the only organelles present. Remember that. Ribosomes are the only organelles present in prokaryotes. And these are not membrane bound. So we'll call it membraneless. They are of small size. And what is the type? 70S type. Is that clear? And the 70S type is further made up of two subunits. One is 30S and then 50S. Is that clear? 30S and 50S. Noted? That's ribosomes. What is the function of ribosomes? Of course, we know they are the protein factories. They make proteins, right? And unlike in case of eukaryotes, oh guys, we'll have to wait for a while. I hope the whole notes are not gone. Yes guys, please tell me, are these points clear so far? Anyone with any doubt? Anyone with any doubt? Yes, please. Anyone with any doubt till now? Any doubt in today's topic till now? And by the way guys, you will have a class tomorrow as well at the same time from 4 to 5:30, and we'll be completing the chapter. Noted? So configuring, oh my god, this configuration is started now. That is not a good news. It will take a while. All right. Anyways, guys, let's have a small discussion here. Class time timings are till 5:30 p.m. So that was the plan from 4 to 5:30, because I have my plus class also at 7 o'clock. So 7 to 9, there will be plus batches that will continue. Is that clear? And also, let me remind every one of you that on 30th of August, coming 30th, we have our first CBT. So get your things ready, get your subscription done, right? Make the payment, download the admit card, do all those things. Get into that feel, get into that proper feel of NEET 2027 as it is going to be, because it will be in the CBT mode. So the first CBT will be conducted by an academy on 30th of August, and please take care of the centers. So those of you who have centers nearby, I would highly recommend you take it in that mode only, rather than just sitting back home and doing it just on your laptop. I know things have been taken care, but get into that routine so that the last day doesn't feel like a hassle. Yes guys, it's 15%. Anything that you would like to ask, we can. Nature of bonding monomers in polymer. Thakur, what are you talking about? We'll talk about all of it. Yes, guys. Flowchart all topics in one slide, ma'am. Helpful, very revision. Vimla, yes. So try to make your notes also in the same manner. Oh, it's done quickly. 100% done. We'll start soon. Yes, guys. Please let me know if there is any doubt. Noted? Yes, please. Yes. Anyone with any doubt so far? We can utilize this time and then proceed. Yes, guys. Anyone with any doubt? Oh, acha, you are Earth Nari. Okay, Earth Nari. Hi. All right. I hope there is signal soon. I'm so sorry. Please listen. Open soon. All right, guys. Yes, please. Phoenix 2.0. Yes, yes, yes. So all the Phoenix students are here too. Structure of protein will be discussed in biomolecules. That is the third chapter that we have to do. That would be the third chapter. Guys, I think there is some issue here. And it's gone. Wait. Let's see. It will. I hope it is still alive. No signal? Doesn't look like a good news. It's not responding. Give me a second, guys. Ma'am, in prokaryotic may or may not be cell wall. Yes, cell wall may or may. See, most of them does have cell wall. Most of them does have cell wall. But most of them does have cell wall. But mycoplasma doesn't have. Okay? There is no signal, guys. By the way, tomorrow's timings are 4 to 5:30. Maybe we'll have a longer class tomorrow. And I'll have to get this checked. That's it. Ma'am, monocotyledonous root. Of course, Thakur. We will talk about monocotyledonous root later on. Anyways, guys, I think we have to wind up now because of no signal here. I'll see you tomorrow. We'll continue with the chapter and we'll be completing the chapter tomorrow. All right. So that is it. I'll see you tomorrow at the same time, 4 to 5:30. And this is complete your backlog series. Master it, not just complete it. And I'll see you in, we'll practice the questions in the Unacademy Learners app. See you tomorrow, same time, and share it with all your friends and bring all your friends here. Bye-bye.